Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing CUET or CUCET entrance exam, MSc entrance exam question paper PGQP 22. It was conducted in the year 2022 and the subject was life science. I have solved only the biology questions. So coming to the first question, which of the following technique is used for tracing change in intracellular location of proteins and metabolites? Chromatography, no, it is used for separation. DNA footprinting, it is used for detection of protein DNA binding specificity. DNA end labeling, it is useful for visualizing small amount of DNA. And pulse chase experiment, it is commonly used technique for studying the synthesis, processing and transport of protein. So for tracing change in intracellular location of proteins and metabolites, you can use pulse chase experiment out of the given options. Then next question. A specific protein required for infecting a host by coronavirus is known as. This question most of you all must be knowing because it has come in the paper. So it is called as the spike protein. Answer is option 3. Then next one. The most common initial step to separate water soluble proteins from insoluble cellular material. So cellular material, you have got a protein inside the cell and it is a water soluble protein. So you want to separate it from the insoluble cellular material. So which step will you commonly use? So the step that you will commonly use is differential centrifugation. Now, usually membranes are composed of lipid protein complexes which have specific densities, and this allows them to be separated from the soluble proteins through differential centrifugation technique. Then next one. The pH at which the net charge of a protein becomes zero under an electric field at a room temperature is known as. This is a very easy question and the answer is isoelectric point. Answer is option 4. The microscope that converts slight differences in the refractive index and cell density into easily observed differences in the light intensity is known as. So such a microscope is known as phase contrast microscope. It converts the slight difference in the refractive index and cell density into easily observable difference in the light intensity. Then next one. Which of the following histone proteins found in chromatin have high lysine content? Now you all know that histones are rich in arginine and lysine protein. Now they have asked for which histone protein found in the chromatin has high lysine content. So it is H1 histone that is having high lysine content in the chromatin. Then next one. Which of the following techniques may be used for identifying a regulatory protein that act by binding to specific sequence of DNA? So southern blotting, it is used only for DNA analysis, northern blotting for RNA analysis, western blotting is only for protein analysis. Now they have asked us the technique that is used for identifying a regulatory protein that acts by binding to specific DNA sequence. So what they meant in the question was which technique may be useful to study DNA protein interactions. So the answer here is southwestern blotting. Which of the following diseases in humans is caused due to expanded microsatellite repeat? So it is Huntington's disease that is caused due to expanded microsatellite repeats. Then next one. Which of the following disease is associated with Philadelphia chromosome? It is chronic myelogenous leukemia that is associated with Philadelphia chromosome, CML. Then next one. A classification system that groups organisms together based on the similarity of their observable characteristic is known as. So first option is phylogenetic system. So it is based on evolutionary relationship among biological entities. They have asked for similarity of observable characteristics. So that is phenetic system that deals with classification of organism based on similarity of their observable characteristics. Phyletic system is, it names groups of organisms according to their evolutionary history. And complement system is what you have learned in your immunology. It is nowhere related to any classification. So next question. A class of immunoglobulin present in the serum which neutralizes toxins, activates complements and crosses the placenta to protect the fetus and neonates. So it is IgG. IgG can cross the placenta. Then next one. Blood group antigen differ in their glycosylation pattern, phosphorylation pattern, methylation pattern, ubiquitination pattern. So blood group, it is ABO system of blood group, and blood group A has A antigen on the red blood cells and anti-B antibodies in the plasma. Now they have asked for blood group antigen differ in their. So they differ in their glycosylation pattern. So the answer here is option 1. Then next one. The replication on template strand always proceeds in 5' 3' direction. If the sequence of template strand is 5' ATCCCT3, how would the correct sequence of newly synthesized strand of DNA be read? So you have to make the complement of this particular strand, and it will be in the 3' to 5' direction. So 3' will be T, 1 minute. So complement of A will be T, of T will be A, of C will be G, G, G and again A. So that is 3' to 5'. So the correct answer here is TAGGA. Then next one. Which of the following condition is not essential to maintain Hardy-Weinberg equilibrium in the population? So large randomly mating population, that is characteristic of a Hardy-Weinberg equilibrium. Small randomly mating population. So that is not essential to maintain Hardy-Weinberg equilibrium in the population. Absence of gene flow, absence of natural selection. They both are essential to maintain Hardy-Weinberg equilibrium. Next one. Bacteria produce an enzyme methylase which play a role in. So methylase, you might have studied it in host restriction and modification, whereby bacteria DNA doesn't get cut by its own restriction enzyme. Why? Because the DNA is methylated at the specific recognition sequence. So the answer here is modification of DNA. Answer is option 1. Then next one. In anaerobic condition, yeast consumes more glucose than in the presence of aerobic environment. This phenomenon is known as, it is called as Pasteur effect. That is inhibiting effect of oxygen on the fermentation process. So they have asked for in anaerobic condition, yeast consume more glucose than in the presence of aerobic environment. Why? So and this phenomenon is called as Pasteur effect, and in Pasteur effect, what is it? It is inhibiting effect of oxygen on the fermentation process. Then next one. Which DNA repair system is affected in Lynch syndrome? So Lynch syndrome, it is the most common cause of hereditary colorectal cancer. Now which DNA repair system is affected? It is the mismatch repair system that is affected and result in Lynch syndrome. Then next one. Which of the following animal has a pseudoseed? So false coelom. So it is found in Ascaricus. Answer is option 1. Which of the following is not a characteristic feature of bony fishes? Presence of paired fins, that is characteristic. Laterally compressed body, that is also characteristic. Presence of cercal tail, that is also a characteristic. Presence of placoid scales, that is not a characteristic of bony fishes. They have cosmoid, ganoid and telosed scales, not placoid scales. So the answer here is option 4. The method which was first developed for gene mapping in bacteria was, so first developed gene mapping technique in bacteria was conjugation in the year 1946. Then next one. The gold standard method used for the detection of SARS-CoV-2 infection is, it is RT-PCR. It was there in papers. So I am not explaining much about it. Then next one. Which of the following phytochemicals produced in plants defend against herbivores? So it is tannins, that are phytochemicals which are produced by plants, and these tannins, they defend against herbivores. Then next one. The rate of oxygen production is enhanced when there is operation of. So in cyclic photophosphorylation, ATP is produced. In non-cyclic photophosphorylation, ATP, NADPH and oxygen are produced. And in oxidative phosphorylation, there is consumption of oxygen. So they have asked for the rate of oxygen production is enhanced when there is operation of. So when there is operation of non-cyclic photophosphorylation. So answer is option 2. Then next one. The primary acceptor of carbon dioxide in C4 plant is phosphoenol pyruvate. Answer is option 1. Then next one. The prokaryotic microorganism which fixes atmospheric nitrogen in symbiotic association under aerobic condition in aquatic ferns. So in aquatic ferns, which is the prokaryotic microorganism which fixes atmospheric nitrogen in symbiotic association with aquatic ferns? So it is Anabaena. Answer is option 3. Which of the following is not a vitamin? Biotin. Biotin is vitamin B7. Choline, it is similar to B vitamins. Histidine is an amino acid. And nicotinamide is vitamin B3. Answer is histidine, which is an amino acid. It is not a vitamin. Then next one. A cloning vector may not contain. So it may not contain a promoter. So answer is option 3. A lambda phage genome has a length of, so it has a length of 42.5 kb. Answer is option 3. Then next one. Chemically synthesized linker molecules containing recognition sequences of specific restriction endonucleases for creation of cohesive ends in blunt-ended DNA fragments are generally. So they have asked what is the size of the linker molecules. Linker molecules are used for what? They contain recognition sequence of specific restriction endonucleases, and they are used for the creation of cohesive ends in what kind of fragments? In blunt-ended fragments during cloning experiments. So the answer here is option 3, that is 6 base pair. Next one. Which one of the following is autoimmune disease? So a disease in which the body's immune system attacks its own healthy cells is called as autoimmune disease. So out of these given options, type 1 diabetes mellitus is an example of autoimmune disease. Then, in which of the following plants the gametophyte does not represent an independent free living generation? That is, gametophyte is dependent on the sporophyte. That is what they have asked. So gametophyte is dependent on the sporophyte in the case of Pinus as per these options. Answer is option 4 and Pinus is a gymnosperm. The next one. Which of the following statement is true about gymnosperms? Albuminous cells in phloem are absent. Albuminous cells are present in gymnosperms. The embryo that grows after fertilization is exoscopic in nature. So it is exoscopic in nature, it is found in bryophytes, not in gymnosperms. Gymnosperms are dioecious in nature, they are monoecious in nature. The embryo that grows after fertilization is endoscopic in nature. That is correct about gymnosperms. So the answer here is option 4. Then next one. Gaucher's disease is a genetic disorder which is associated with. So Gaucher's disease, it is associated with the lysosomes. So the answer here is option 4. Hematopoietic stem cells are found in. So hematopoietic stem cells are found in the bone marrow. Which of the following vaccine does not provide lifetime protection? Polio provides. Chickenpox. Chickenpox doesn't provide lifetime protection. Chickenpox vaccine doesn't provide lifetime protection. They have asked for the vaccine, not for the disease. Smallpox also provides and MMR, that is measles, mumps and rubella vaccine. It also provides lifetime protection. The next one. The formation of endosperm before fertilization is a characteristic of. So it is a characteristic of the gymnosperms. Answer is option 3. Heterospory is not found in. So heterospory is formation of two types of spores. Now Pinus, Cycas and Selaginella, they produce microspores and megaspores. Whereas Pteridium, it doesn't produce. It is homosporous, produces only one type of spores. So the answer here is option 4. The next one. Diagnostic test used to detect human deficiency virus. So which diagnostic test is used in to detect HIV? So it is ELISA that is used. Vidal is used for typhoid. So ELISA technique is used to detect human deficiency virus. Then, which one of the following antibody can cross placental barrier? Again the same question. It was asked just 10-15 questions previous. So it is IgG that can cross the placental barrier. Then trypan blue assay is used for. So trypan blue, it is used to color dead tissues or cells selectively. So it will color dead tissues. So it can be used for counting of animal cells. So answer is option 1. Then next one, match the following. So enzymes and their function. Histone acetyl transferase. Histone acetyl transferase is for chromatin remodeling. Chromatin remodeling, it is a mechanism for regulating eukaryotic gene expression. And in this what happens is the tightly condensed DNA portion, they become accessible to various transcription factors and components of DNA replication, and thereby it will regulate eukaryotic gene expression. If it is in the condensed state, the transcription factors will not be able to bind and the gene expression will not be there. Now once it is decondensed, or what happens is the DNA sequence will be available for the transcription factors to bind and expression of the gene happens. So histone acetyl transferase is chromatin remodeling. A2 is there only in one option, so that itself will be the answer. But anyway, we will see the others also. Cyclin dependent kinase. Cyclin dependent kinase is for cell cycle regulation. Alpha antitrypsin. Alpha antitrypsin is for emphysema. and topoisomerase is involved in DNA replication. So if you know one option correctly, that is A2, you can happily mark the answer. Then next one. Again match the following. List 1 includes animals and 2 includes their larva. So out of these, which is the correct option? Annelids. Annelids is Aminocetus larva. So A, 1 minute. Okay, annelids is Trochophore larva. Answer is A2. Insects it is caterpillar. Mollusks it is veliger, and cyclostomes it is Ammocetus. So the answer here is A2, B4, C1 and D3. So the answer is option 3. Then next one. Epinephrine in liver. Epinephrine in liver and its function in the liver, it is 2. Increased conversion of glycogen to glucose. A2. A2 is found only in option 4, so that is the answer. And we will see the others also. FSH. FSH is increased synthesis of estrogen. Vasopressin. Vasopressin is for reabsorption of water. And epinephrine in skeletal muscle, it is for conversion of glycogen to glucose 1-phosphate. So the answer here is option 4. Then next one, they have given the nitrogenous bases and the deaminated product. So cytosine on deamination, it will give rise to uracil, A3. 5-methyl cytosine on deamination, it will give rise to thymine, so B1. Adenine on deamination will give rise to hypoxanthine, C4. And guanine on deamination, it will give rise to xanthine, so D2. So the answer here is A3, B1, C4, D2, and the option is option 2. Then next one. They have given the compound and its function. So dolicol. Dolicol, it is sugar carrier. Dolicol phosphate. If I remember, it is there in endoplasmic reticulum. So dolicol phosphate, it is for sugar carrier. Then vitamin K. Vitamin K is blood clotting factor. It is needed for blood clotting, vitamin K. Vitamin E is for as an antioxidant, and warfarin, it is anticoagulant. So the answer here is A4, B1, C2 and D3. Then next one, cell type and their function. So macrophages. Macrophages are involved in phagocytosis. A2. B cells are involved in antibody production, B1. So A2, B1 means option 2 will be the answer. We will see C and D also. Cytotoxic T cells. Cytotoxic T cells, they interact with infected host cells, and helper T cells, they interact with macrophages and secrete cytokines. So the answer is option 2, A2, B1, C4 and D3. Then next one, they have given the name of scientists and their discoveries. So Avery, MacLeod and McCarthy, they are involved in chemical basis of heredity, that is DNA as the basis for heredity. Then Edward Jenner, he was with vaccine, smallpox vaccine. And Erwin Chargaff, he was with the law of equivalence of bases in DNA. And Seymour Benzer was involved in complementation analysis. So the answer here is option 4, A3, B4, C2, D1. Then next one, Given below are two statements. Statement 1. The application of herbicides to kill unwanted plants is widespread in agriculture. That's correct. DCMU act by blocking electron flow at the quinone acceptors of photosystem by competing for the binding site of plastoquinone. That is the action of DCMU, which is also a herbicide. So, in the light of the above statement, choose the correct answer from the options given below. Both statements 1 and 2 are correct. Yes, that is correct. Then next one. In prokaryotes, all types of RNAs are transcribed by the holoenzyme that consists of several polypeptides, 2 alpha, 1 beta, 1 beta dash and 1 sigma. Statement 2. The start signals on DNA are called promoters and represent the initial binding site for RNA polymerase. So here also both the statements 1 and 2 are correct. Then next one. Given below are two statements. Statement 1. Glycolytic pathway is the only route available for oxidation of sugars in plant cells. Not only glycolytic pathway, you have pentose phosphate pathway also. So first statement is not correct. Then in statement 2, in pentose phosphate pathway, the complete oxidation of one glucose 6-phosphate molecule to carbon dioxide result in the production of 12 NADPH molecules. That is correct. So statement 1 is incorrect, but statement 2 is correct. Answer is option 4. Then next one. Telomeres are sequences at the end of eukaryotic chromosome which helps in stabilizing the chromosome. That is correct. Telomeres with multiple repeated sequences in the form of TTA GGG times in humans. That is also correct. So both the statements 1 and 2 are correct. Then next one. Given below are two statements. In agarose gel electrophoresis, the large pore size of the low concentration gel make possible the separation of very large molecules. That is correct. Then in agarose gel electrophoresis, the small pore size of the high concentration gel make possible the separation of large molecules. It is not separation of large molecules, it is separation of small molecules. So statement 2 is incorrect. So answer is option 3. Statement 1 is correct, but statement 2 is incorrect. Then next one. Statement 1. Mycorrhiza is a symbiotic association between cyanobacteria with root of higher plants. It is not cyanobacteria, it is fungi with root of higher plants. So statement 1 is incorrect. 2. Mycorrhizal association is important for nutrient acquisition in plants under limited condition. That is correct. In mycorrhizal association, phosphorus is supplemented, if I remember it. So the answer here is statement 1 is incorrect, but statement 2 is correct. Then next one. Given below are two statements. Apoptosis, that is programmed cell death, is a genetically regulated process occurring in specific tissues during development and disease by which cell destroy itself. That is correct because you can see that in the case of regeneration, in case of animals. Then apoptosis is regulated by intracellular signals generated from many secreted cells, cell surface, protein hormones as well as many environmental stresses. That statement is also correct. So both statements 1 and 2 are correct. Then next one. In C4 cycle, fixation of carbon dioxide by the carboxylation of phosphoenol pyruvate in the mesophyll cells result in conversion of malate or aspartate. So that's correct. The transport of C4 acid to the bundle sheath cells lead to decarboxylation and generation of carbon dioxide, which is finally reduced into carbohydrates via Calvin cycle. That is also correct. So both statements 1 and 2 are correct. Then next one. Establishment of mammalian cell culture involves the following step. Choose the correct answer from the options given below. Culture of primary cell, subculture of primary cell, separation of cells from tissues by mechanical or enzymatic method. That is the first thing that you have to do when you plan to establish a mammalian cell culture. Culture of secondary cell, subculture of secondary cell. So after this separation of tissues by mechanical enzymatic method, you get the primary cell. So you have to culture of primary cell is the second one. Then subculture of primary cells, that is third. Then culture of secondary cells, 4. And subculture of secondary cell, that is 5. So the answer here is C, A, B, D, E. So that is option 1. Then next one. The steps involved in bacterial translation are activation of amino acids, initiation of polypeptide synthesis, aminoacylation tRNA, peptide bond formation, termination of polypeptide. So activation of amino acids, that is the first step. Then comes aminoacylation tRNA, 2. Then comes initiation of polypeptide synthesis. Then comes for peptide bond formation and finally termination of polypeptide. So the answer is A, C, B, D, E. Answer is option 1. Then next one. The sequential steps involved in prophase 1 of meiosis are chromosome condensation, terminalization of chiasmata, formation of tetrads of bivalence, pairing of homologous chromosomes, genetic recombination. So in the prophase 1 of meiosis, there is chromosome condensation in the beginning. Then pairing of the homologous chromosome take place. Then a formation of tetrad or bivalence or 4 strands that happens. Then you have got genetic recombination, packeting stage, remember. And then terminalization of chiasmata. That is fifth one. So the answer here is A, D, C, E and B. So that is option 3. Then next one. Arrange the following discoveries in increasing order. So first one who was the discovery of origin of species by Charles Darwin. So that is natural selection. So Charles Darwin published a book on origin of species. So that is first one. Then comes Gregor, Johann and Mendel with the principles of genetic law of segregation, independent assortment, purity of gametes, everything. Then later Friedrich Miescher identified nucleosine. So that is the third one. And then comes Oswald Avery identified DNA as the transforming principle. After Griffith experiment, Avery has done that. So that is the fourth one, and the fifth one will be Watson-Crick deciphered the DNA structure. So the answer here is B, A, D, E, C. So that is your option 3. Then next one, arrange the following in order of their action. So G1 cyclin CDK complex, this is in the case of your cell division. APC-CDH1, S-phase cyclin CDK complex, MPF, APC-CDC20. So the first one will be G1 cyclin CDK complex A. Then comes S-phase cyclin CDK complex. So first one will be your G1 phase. Then comes the S-phase cyclin, that is S-phase cyclin CDK complex. Then comes your mitosis phase, MPF. MPF will be maturation promoting factor that promotes entrance into mitosis. Then comes your anaphase promoting cell, that is C. APC. That will be E, that is APC CDC20, and finally APC CDH1. APC is anaphase promoting complex. So the answer here is option 1, that is ACDEB. Then next one, identify the correct order of signal transduction pathway. So signal transduction, binding of ligands to cell surface receptors, termination of signal, generation of intracellular second messenger, intracellular signaling cascade, effector, response. So first will be binding of ligands to the cell surface receptor. So with that itself option 2 and 3 can be cancelled out. We have to see for option 1 and 4. Binding of ligands to cell surface receptors. A is correct. Then comes generation of intracellular second messenger. That will be second one. Then intracellular signaling cascade. That will be third. D, E, effector response. Then it will be fourth. And finally termination of the signal. So A, C, D, E, B. A, C, D, E, B is your first option. So answer is option. Then next one. The nucleoplasmic index, where the nuclear mass and cytoplasmic mass of the cell are in the state of optimum equilibrium, depends upon. So it depends on nuclear volume and cell volume. So A and B only. Answer is option 1. Then next one. Identify the inhibitory neurotransmitters from the following. So inhibitory neurotransmitters. Glutamate, aspartate, cysteine, glycine, GABA. So one is GABA, E will be there, and the other is glycine. These two are inhibitory neurotransmitters. So D and E, that is your option 4. Then next one. The function of a gene depend on the expression of. So function of a gene, it depend on the expression of RNA as well as proteins. That determine the function of a gene. So answer is B and C only, that is option 3. Then next one. Identify the features of cancer cell. Rapid cell growth and division. That is correct. That is a feature of cancer cell. Reduced cell growth and division. No. Loss of normal control of cell division. That is also correct. With that itself your answer has come out. A and C only. That is option 3. Reduced angiogenesis. Angiogenesis is increased in cancer cells and utilize glucose at reduced rate. So they utilize glucose at increased rate. So A and C only. The next one. Identify the methods to measure apoptosis in vitro in mammalian cell cultures. So MTTT assay is used for measuring apoptosis in vitro in mammalian cell cultures, as well as NMR is also used. So the answer is A and C only, that is option 3. Then next one. Identify the tumor suppressor genes from the following. So out of the given genes, which is the tumor suppressor gene? That is P10 and P53. They both are tumor suppressor genes, whereas others are not. C and E. So the answer is option 2. Then next one. Which of the following organizations are involved in co-vaccine production in India? So ICMR, NIV and Bharat Serum. Sorry, Bharat Biotech. So C and D are the answer. Answer is option 4, C and D only. Then next one. Given below are two statements. One is labeled as assertion A and the other is labeled as reason R. Okay. So assertion A. Bateson and Punnett showed that the linked genes do not obey Mendel's law of independent assortment. That is correct. Then next one, reason. The dihybrid test cross ratio for closely linked genes produced 50% parental and 50% recombinant type of progeny. That is wrong because it is not 50% recombinant type of progeny. It is less than 50% of the recombination type. So R is not correct. So A is true but R is false. Answer is option 3. Then next one. Given below are two statements. One is labeled as assertion A and the other is reason R. So assertion A. Oligosaccharides with unique structures are components of a variety of glycoproteins and glycolipids on the outer surface of plasma membrane. So that is correct. The influenza virus infects animal cell by binding to the cell surface glycoprotein as a first step of infection. That is also correct. So A and R are true and R is the correct explanation of A. So option 1 is the next one. Here also assertion and reason. So assertion is each dideoxynucleotide used in Sanger's method of DNA sequencing can be linked to a fluorescent molecule that gives all the fragments terminating at that nucleotide of a particular color. Yes. So if you label the dideoxynucleotide with the fluorescent molecule, all the fragments terminating with dideoxynucleotide will give that particular color. So in that way you can identify which is A, which is T, which is C, which is G. Then the DNA sequence is read by determining the sequence of color in the peak as they pass through the detector. That's what I told just now. So assertion A and R both are correct and R is the correct explanation of A. So the answer here is option 1. Given below are two statements. Okay, again assertion and reason. Polyploid species of common bread wheat have evolved by hybridization followed by chromosome doubling in interspecific hybrids by treatment with colchicine. Okay, that is correct. Then colchicine induces gigantism in yellow polyploid species of bread wheat. In the light of the above statement, choose the correct answer from the options given below. Colchicine induces gigantism in yellow polyploid species of bread wheat. That is not correct. Colchicine induces polyploidy by preventing the segregation of chromosome. That is why here chromosome doubling has happened. Polyploid species of common bread wheat have evolved by hybridization followed by chromosome doubling in interspecific hybrids by treatment with colchicine. So A is correct but R is false. Answer is option 3. Then next one. Again assertion and reason question. The LAC-Z gene is present in PUC cloning vectors as a selectable marker by which recombinants may be selected through insertional inactivation of LAC-Z gene. That is correct. IPTG act as a gratuitous inducer of LAC-Z gene due to which recombinants produced in PUC vector may be selected by blue-white screening. That is also correct. Here what happens is in PUC cloning vector you have got a LAC gene. Now this LAC gene, it will have multiple cloning site in it. So because of the presence of multiple cloning site, you can insert any gene of interest. So once you insert a gene of interest in the LAC gene, LAC-Z gene will get inactivated and you will get white colonies. So your recombinants will be white colonies and blue colonies. That is because the LAC-Z gene doesn't have the insert in it, will give rise to blue-white colonies, and IPTG is acting as the inducer of LAC-Z gene. So the answer here is both A and R are true, and R is the correct explanation of A. Answer is option 1. Then next one, they have given group of organism or plants and they are grouped as. So bacteria and blue-green algae, they come under Monera, A3. There goes your answer, that is D. 4. Bryophyte and pteridophyte, they belong to embryophyta. Pteridophytes and gymnosperms, they belong to tracheophyta. Angiosperms and gymnosperms, they belong to spermatophyta. So the answer here is option 4. Next one, Rauwolfia serpentina and its economic importance. So economic importance of Rauwolfia serpentina is that it is involved in blood pressure treatment. So A3, Vinca rosea is involved in cancer. B4, Schizygium aromaticum, it is in toothpaste. And Azadirachta indica, that is your neem tree, it is involved in insect repellent. D1. So the answer here is option 2. A3, B4, C2, D1. So that's it. Thank you for watching my channel. Please do like, share and subscribe. Then I would like to tell you one more thing, that I have uploaded 2021 question paper also of this life science question paper conducted in the year 2021. You may go through that also in my channel. So thank you for watching.